All right, we're going to continue with morphogenesis, talking about first morphogens, how they create the overall body plan, which is the central event in embryology. We're going to talk about how a cell's cytoplasmic chemicals shape its fate and that of its lineage. We're going to talk about organizer cells and how they use inductive signals to alter the fate of other groups of cells. And then we'll talk a bit about hormones and how they shape organ development both before and after birth. So how does a body come about from a single fertilized zygote? We know a lot based upon both classic and modern morphogenic studies that sometimes just use cutting and pasting of developing embryos, as well as cutting-edge molecular work. And this cutting-edge molecular work, as well as the cutting and pasting, help reveal the formation of the body axis and body parts. And they show that scalpels are still really important in a lot of this work. So animal clades are distinguished by their body plans. We've talked about that for the last several lectures. And it is chemical signals or morphogens that create the body plan, this top, bottom, front, back, whatever, which is the central event in embryology. So a morphogen is just any chemical signal within the body. And morphogens induce change. So the process of morphogens inducing change is called induction. And that's when a chemical signal or morphogen from one cell group will alter, the plus or minus just means increase or decrease, things like the rate of cell division, or the rate of cell movement, or the rate of gene expression in another group of cells. So a chemical signal from one group of cells might say, increase the rate of cell division here, but not over here. Or it might tell cells to migrate towards this signal and away from that signal. Or it might tell genes in certain cells to turn on at a certain time or in the presence of a certain chemical, or off at a certain time or in the presence of a certain chemical. So what you're seeing here is the process of gastrulation, that invagination of the blastula to form the gut. And so one of the great questions in developmental biology was what causes that process? What causes the formation of the digestive tract? And it turns out that two individuals, Hans Spemann and Hilda Mangold, simultaneously worked on figuring out what this organizer group of cells were. People had long thought that somewhere within the developing blastula, there must be an organizing group of cells that sends out a signal that tells other cells to start that process of invagination. So they did a couple of really cool experiments that kind of nailed that there is in fact an organizer that is inducing the movement of the cells that you just saw. Well, first off, in a developing embryo, on the opposite side of where the sperm enters the egg, something forms called the gray crescent. And this forms after fertilization. In the little images you see here, the yellow part on the bottom is actually the yolk, and the blastula is the gray and light gray part on top. The light gray part is the gray crescent. And that is actually where this gastrulation process begins. And so they thought that that might be an important organizing set of cells in this process of gastrulation. So let's see what they did. They took two frog embryos and they took a very fine string. In fact, I think it was an eyelash from a human. And they divided the developing embryo in two in such a way that each of the two halves received part of the gray crescent. And what they said was if the gray crescent is fundamentally an organizer that induces gastrulation, then both of the embryos should in fact develop normally. And that's exactly what they saw. The embryos might have been a little bit smaller because they had less yolk, but they were functionally normal. Then what they did is they took that embryo and they rotated it 90 degrees. And they did the same thing so that they divided the embryo in two, but now only one side received gray crescent and the other side received none. And what they got when they did that was one fully functional tadpole gastrulation proceeded as normal. But in the other one, it was just a lump of tissue. There was no gastrulation. And that's because they said that there was no organizer in that other lump of tissue. So without the organizer to induce gastrulation, no gut forms and development does not proceed as it should. So they continued with this experiment with a transplantation experiment. And what they did here was they simply took two different embryos and they took the region of the gray crescent that forms called the dorsal lip, and they transplanted it from one embryo to the other side of a second embryo. So here we have an albino salamander or newt and a pigmented salamander or newt, and they took a section from the pigmented newt that they thought would induce gastrulation, and they transplanted that on the other side of the gray crescent and dorsal lip of the albino embryo. So now it has two dorsal lips, two gray crescents. And so what they thought was if there are two separate organizing signals, then we should see gastrulation proceed in two separate spots. And that's what they did. And that's what they got. They actually got two gastrulation sites, two guts developed, and they got two developing embryos. Now here, this is a zebrafish embryo that you see, but it's the same process. And here's the actual transplantation experiment. And again, they're using an eyelash, I believe, to cut a section off of one developing embryo and to put it onto another. And realize that these developing embryos are incredibly plastic. So you can just hack off a chunk of cells from one, attach it onto another, and it's almost like it has these magical healing properties that will just sort of be assimilated into the new developing embryo. It's pretty remarkable. So these two experiments, the splitting of the embryo and the transplantation experiment, told them that yes, there most certainly is an organizing signal, and we know where that organizing signal is. And we see similar things all over the animal kingdom. So here's an example in vertebrates. If you want to understand things like what makes a top versus a bottom, like a back versus a belly, there is a gene called BMP4 that helps to ventralize vertebrate embryos, create a belly versus a back. And BMP4 stands for bone morphogenic protein. So this is critical in making things like the spinal cord. And vertebrates also make something called cordon, and there's a process that allows us to see where genes are actually being expressed. And we can visualize that cordon is being expressed up here in this dark part of the embryo, but not in the bottom. And Xenopus is a frog. It's a model organism frog used in development. So the thing with cordon is that it actually blocks the ventralizing properties of bone morphogenic protein. So wherever there's cordon, bone morphogenic protein doesn't function because it blocks the BMP receptor. And because of that, it creates this dorsal ventral body plan. You have a clear differentiation between areas where bone is produced versus where bone is not produced. What's crazy is that we see an orthologous gene in invertebrates. And remember, orthologous genes are genes that are similar, at least in function, in two different lineages because of a lineage splitting event. Invertebrates and vertebrates shared our most recent common ancestor hundreds of millions of years ago, so it makes sense that the ventralizing properties of the two lineages are somewhat different. So they make an ortholog that blocks a BMP ortholog because they have a common ancestor. So a slightly different process, but that process is similar because of common ancestry. Another area where we see induction is in the production of the lens of an eye. So during development, we see these bulges in the anterior region of an organism, and we see two different tissue types. We see this green tissue that forms something called an optic cup, and then we see this invagination of the blue ectodermal tissue forming something called the lens that we're all familiar with. So another famous experiment by Hans Spemann was he was interested in who or what was inducing what. Was the optic cup tissue inducing the lens to form or vice versa? So the first thing he did was he removed that optic cup tissue from a developing embryo and watched what happened, and no lens formed. So that's pretty good evidence that there must be some organizing signal inducing the lens to form because of the optic cup. He also said, well, look, if I take the ectoderm that produces the lens and there's an organizing signal in there and I move it to another part of the body, then it should develop an optic cup. And when he did that, nothing formed, period. So that was more evidence that there's no signal within that ectodermal tissue producing the lens. And then finally what he did was he transplanted the optic cup to the abdomen of an organism and saw that a lens formed. So that signaling property of the optic cup, even when moved to a different part of the body, not the face, still produced a lens, which is pretty remarkable. And this is what it looks like right here. So they transplanted the optic cup to a region of the tail, and an actual lens formed there because there's ectodermal tissue there. Now, that's not a functional eye because other aspects of the eye needed to see are developed by other tissues and other organizers, but the lens itself actually forms there, which is pretty remarkable. Now, we've talked about regulatory genes and master control genes. For the development of the lens and optic cup, it's PAX6. And we talked about PAX6 earlier in the semester when we were talking about orthologous genes. And this is a classic example because we see orthologs in humans, in mice, in zebrafish, even in fruit flies. And this cascade that you see to the left kind of shows you how these regulatory genes work. What you see is the gene itself is the little straight line. The protein product is the little Pac-Man looking thing produced. And what you see is that PAX6 doesn't actually produce the functional genes of the eye, which are I1, I2, I3 towards the bottom. It produces a regulatory protein that when it lands on other downstream genes, either turns them on or turns them off. So you have the master control gene that lands on eye tissue gene number 1. It lands on eye tissue gene number 2. That produces an eye tissue gene number 2, which is blue, which also has to land on eye tissue gene number 1. Of course, you don't have to learn this, but understand that there are really, really critical regulatory mechanisms in place so that only certain tissues are produced in certain places. That's the whole idea of regulation and regulatory genes. How old is PAX6? It's a really old gene. We see PAX6 orthologs and basically the ancestor to all animals. And so when you ask, why do animals have so many different versions of an eye or a lens? It's because they all share a similar version of that gene, the PAX6 gene. Okay, let's take a break before we talk about additional induction in different tissues.